and 982 we've currently sequenced. And in these patients, what you can see is here's someone by standard karyotyping had an inversion 16, which was picked up as the gene fusion that it's supposed to be. But we also picked up variants in both NPM1 and FLT3-ITD that were completely concordant with clinical data that was available. But in addition, in a very small set of patients, we've also already picked up a new mutant here. And we don't know whether this deletion acts the same way as duplications that are seen in these other patients, but it's in the same region where the activity of the protein is increased when you have it. And so the potential here is to pick up more than we currently do with standard testing, and hopefully better stratify patients. So the way the process begins is that samples in a prospective study would be that samples would arrive at the cancer genetics lab. The sample would be accessioned, DNA and RNA would be extracted and delivered to the CDG, at which point library preparation would be conducted and the DNA would be sequenced. And we're looking to eliminate potentially one step here. This sequencing quality is then first approved by the sequencing group, but then goes through a very rigorous QC through the bioinformatics group. And because all of these take quite a bit of time, what we've had to do is to try and develop a parallelization of the informatics so that the quality control is going on at the same time that we're looking for the mutants that exist within these patient samples. And at the end of that, we will be delivering a report of the mutation found. So with the normal process, sequencing is performed. The technologist will look at the data, will highlight where there are potential mutations or not, where we will be looking for one. And then this is passed on to a clinical scientist or a pathologist for sign-out. The data that comes out of the next generation sequences is actually the raw data are primary image files, which are then converted to streams of text, which is very difficult for a technologist to examine. And so this basically shows what a current test would look like. This is an analog of a Sanger sequencing data chromatograph, where you can very quickly look at the quality, because this is real signal, this is noise down here. You can see where there's a mutation because there are two different colors. But what you see in a viewer, if you were now to take that text string that I showed you before and convert it into a viewer, you can now have the software highlight where there's a discrepancy in the sequence, and now a technologist can go in and look at this data and determine what the quality of that call is and whether it should be reported on or not. And so this is just a high-level view where a technologist will scan through and say, these are the gray bars are what's sequenced, there's no sequence here, and here's where a variant is called, and then this can be examined with more granularity. So I've talked to you a little bit about the development of the clinical test, but integrated with developing the test is also an economic evaluation and the ethical and privacy issues surrounding genomic testing. Now, just to clarify, we are not looking at, in this project, constitutional DNA. We're not looking, for instance, at a blood sample of normal DNA. We're looking at tumor tissue. So one could argue that within any variant found within the tumor tissue may or may not reflect what's present in the host genome. And so I think by using this approach, we could argue that we're only going to look at things we know are relevant to the disease, and whatever else is there, we cannot spend the effort and time to verify because it's only coming from the tumor. It has no impact on clinical management. And so we were trying to get away from finding things like Huntington's disease or BRCA1/2 mutations or other things that are CF mutations quite commonly, cystic fibrosis mutations, that may be found in this data set. So I am not a health economist and I can only repeat what's on this slide, but in essence, the the health economist Aly Karsan, working with Stuart Peacock, has looked at a conceptual model, is now populating the model, and then will go on to do a validation study and determine the value of the information that's determined, that's derived from that, and this will be validated with published outcomes, but hopefully we'll be able to then apply that data to the current study. Peter Chow White is looking at the ethical and communication issues associated with genomic testing, and his three major points of investigation are to look at the practices of producing and managing patient genetic information in the lab in a clinical context currently, to investigate the risk to the individuals and groups of patients involved, and importantly to investigate the regulatory framework that currently govern handling and management of personal genetic information. And I can tell you we're a lot further behind than the US in this area, and so the hope is that in the context of this project, we will also be able to educate the regulatory bodies as to how to proceed. And at the end of that, the idea is to develop a model that balances the benefits and the risks in healthcare settings for patients. The outcomes, as Peter has defined them, is to integrate the findings into a set of guidelines and best practices for clinical and research scientists as well as clinicians, so that will enable us to use this data and enhance care, again, in the setting of protecting patient information. And as part of this project, one of the first things that will be done is to develop an ethical submission to the Research Ethics Board at the BC Cancer Agency to conduct a prospective study in the coming year. And so the question then becomes, the type of information we're generating is complex. It's not necessarily accessible to, and I'm not even talking about the general public, but even to most clinicians anywhere. And so we have to have a way to transfer this knowledge to the users of the data. And the users include clinical labs, so the technologists need to be educated, and I've talked a little bit about that. Pathologists through the province will be educated through this project. And then the clinicians that will be receiving the data will be educated through the Leukemia BMT program of BC. This is where all the stem cell transplantation, or allogeneic stem cell transplantation occurs in BC. And there are a group of hematologists who meet on a monthly basis, who develop guidelines and then disseminate this through our website. And so this project will be used to inform the community physicians as well. And then through contacts at SFU as well as at UBC, various members of the team are faculty at UBC will be able to then transfer some of this as well through the academic setting. Nationally and internationally, a number of us are involved with clinical trial leaders in the US and in Europe, and we will be organizing a meeting to go over the data that you generated, to examine the potential of developing a validation study that involves multiple centers. As I've already mentioned, the Genome Science Center is already part of the TCGA consortium. And a number of us are also involved in a pan-Canadian group engaged in AML clinical diagnostic trials, which will be another mechanism to disseminate this data. And so the timeline is short. It's only 2 years. The first year will be a retrospective study for all three parts of the project: the test development, the decision tree, the economic, ethical decision tree, and the conceptual economic model. We plan to begin a prospective trial in the summer of this year to then validate our findings. So this is just a concept that we're developing for the CBG. which is that in the end we want to deliver a clinical report based on genomic testing. To do that, we need clinically validated genomic tests, and to get a clinically validated genomic test actually takes a lot of effort. So it requires the development of the test, needs the validation of predictive tests as I described for AML, but also screening tests as I've described for BRCA1/2 testing. So this gives you a result. It says there is a mutation or not in a certain area. But we need to be able to interpret that data. And so that requires a whole other investment of informatics where we need to collect data, not just locally, but globally, and to warehouse and meta-analyze that data so that we can predict what a variant means, what a mutation means, so we can predict whether a drug is going to be efficacious or not, so we can predict what the likelihood of an adverse event would be if a certain drug would be used in the context of genomic information. And so all of this is really dependent on clinical informatics, and I'm purposely not using the term bioinformatics because there are very, very clear clinical requirements in order to deliver both the test and the interpretation of the test. And so we have a team built around looking at both aspects of this so that we can deliver a clinical report that has value. And so there are a number of people involved in this project. The Center for Clinical Diagnostics, the key personnel are listed here. The Cancer Genetics Lab personnel are listed here. But there are scores of other people behind us who are actually generating all the information that I've shown you here. Thank you very much.